The University of Michigan has often blazed a trail in advances in medical science. Researchers here performed the world's first successful lung removal. And they pioneered the emergency use of angioplasty. And they discovered the gene responsible for cystic fibrosis, just to name a few. Now a new generation of researchers are working on new tools and therapies to help cure some of our most intractable diseases. And someday, the life they save may be yours.
Go ahead and look at me. Shut your eyes as tightly as you can, okay? Don't let me open them.
When I received the diagnosis of a fatal disease, you end up re-examining all of your decisions, your life, what you do with it, and where you've gone, where you've been.
6 months ago, Bob Shaney was diagnosed with amyotrophic lateral sclerosis, better known as ALS, or Lou Gehrig's disease.
Yeah, definitely. It's nonstop.
Can you feel those?
Nonstop, yeah. So I think, the very first few days was a period of shock and intense grieving. So there's a lot of atrophy in that hand.
Yeah,
you can see it right
really
there.
is.
The disease is part of my life every single day, every single hour. It's part of what I live. It's affected my wife and I in fundamental ways.
Don't let me do that. Good. Don't let me come down. Give me absolutely all you got. Okay.
Yeah, it can happen.
It's the courage of patients like Bob that gives direction to the research efforts of Dr. Eva Feldman.
Are you able to take your thumb and first finger and do like that?
How do you know where to start? How do you know what questions to ask?
So my questions come from my patients. So I'm, as you know, an MD, PhD, and I really cherish both pieces of that occupation. But the questions that I do as a PhD all come from my MD life.
Dr. Feldman is one of the world's leading biomedical researchers and director of the Alfred Taubman Medical Research Institute. She gave me a tour of her lab, part of the Consortium for Stem Cell Therapies, where some very innovative scientists are collaborating in search of cures.
I enjoy the space. As you can see, we have all open laboratory space, which has been very conducive to interacting.
Finding new therapies can often feel like swimming upstream, so maybe it's little wonder fish have been enlisted in the effort.
One thing that we've done, and Stacy Sikowski has helped us with this a great deal, she's a postdoctoral fellow in my laboratory, we've actually hooked the ALS gene onto a marker that makes all the zebrafish who are affected with Lou Gehrig's disease green. So, we can do a neurologic exam on these fish. In a way, the way we examine a patient, we can see how quickly these fish swim.
The fish with the green marker have ALS. They swim more slowly as the disease progresses, so therapies can be evaluated on how long they keep the green fish swimming normally.
The camera is tracking the movement of the fish. The fish are pseudo-colored yellow, and their distance that they're moving is the blue line that's following them behind. And what this software can do is it can actually record those distances and translate it into a velocity for how fast the fish swim, which is correlated with the muscle strength or muscle weakness or degeneration that would occur in ALS.
So these fish become weak, have a progressive weakness, the ALS fish, just like what we see in my patients in our clinic.
Why take the time to develop gene markers that turn fish green or software to track their speed? Because testing with zebrafish has the potential to fast-track new cures.
This is our zebrafish facility, and what we are doing here is breeding many animal models of disease here in zebrafish. Jim Dowling, who's a pediatric neurologist who works with us, was really his invention, his idea to begin to use the zebrafish.
Where did that idea come from?
Well, one of the great things about the building that we work in is that there's lots of other investigators who do other kinds of research, and they recommended that we try the zebrafish.
What's so nice about using the zebrafish is they are permeable to molecules and to therapies. So for ALS or for muscular dystrophy, for example, we can take the fish, put it in a small well, and put a potential therapy, and then see if that helps the fish retain its ability to swim.
Why are the fish so much more suitable for your work?
Faster, much less expensive, and volume. We can screen, for example, 100, 200 drugs in a few weeks, and that would take years if you were using a primate, or even years if you were using mice. So this is going to help us fast-track therapies.
So how many spaces do you have here?
Oh,
The efficiency of testing on zebrafish allows Dr. Feldman's team to quickly zero in on the most promising therapies to pursue. And that's only one of the many collaborative approaches being taken in her lab.
I mean, I've just shown you some zebrafish, and we really take those findings that we have in the zebrafish, we transfer it here to the computer. And so what we have here is we have 4 individuals who work here, and we actually call this our geek bay because they all take our scientific findings and using new novel methods and programming of the computer can ask and answer questions that we can't easily do at the bench.
Such as when Dr. Adam Schuyler constructs an atom-by-atom map of the surface of a single protein.
There are over 100 different mutations that are known on this protein that cause ALS. By looking at these different locations here that change in the disease state, we can then model the dynamics of this protein, and those dynamics are what ultimately drive biological function.
Does this also help you project how the disease may progress?
Yes, with this modeling approach, one of the predictions we can pull up are new target locations, especially on the surface of this protein, that are changing the most according to these different mutant forms. And these are very natural targets for therapeutic design.
Whether it's using zebrafish to sift through hundreds of potential therapies or painstakingly mapping microscopic genetic mutations, the hope this cutting-edge research provides is something Dr. Eva Feldman can take directly to her patients.
You're in a really interesting position in that you are a scientist, a researcher, and you're also a clinician.
Yes.
So you are...
I'm a bridge.
You're a bridge.
I'm a bridge.
What is that like for you?
I really have the best life of anyone you will ever meet. I truly mean that, because I get to go see my patients in clinic and I get to tell them what I'm doing here. I get to go there and say, you know, I am trying very hard to understand what causes your disease and to develop new therapies for your disease. My best hope is that we can use stem cells for the treatment of ALS and that it will minimally stop the progression of the disease if we use them early, and even potentially maybe even help people become better with the illness, something that we'd never even hoped for before. So in the best of all worlds, in the short term, that's what I see. And I don't want to think 10 years out. I want to think 10 months out for my patients.
That sense of urgency isn't lost on people facing the day-to-day reality of currently incurable diseases.
How has your diagnosis changed or developed your perspective on stem cell research?
Yeah, certainly with someone in a condition like mine, parent of 2 young children and husband, I think from my own perspective, gives me and my family some hope that an effective treatment may be not too far out in the future.